I want you to picture a pretty terrifying medical scenario.
Yeah, it's a grim one, honestly.
Right. So you're in your early 20s, you're perfectly healthy, just, going about your normal life. And then over the course of just a few weeks, your central vision begins to just fade to gray.
And it's not like an injury, right?
Exactly. It's not painful. It's not an eye injury. It's happening because deep inside your optic nerve, at a totally microscopic level, a tiny string of genetic code is carrying a typo.
A very specific, very devastating typo.
Yeah. But, and here's the crazy part, this isn't a typo in the main DNA of your cell's nucleus. This typo is inside your mitochondria.
The power plants.
Right. And when those tiny cellular engines fail to produce energy, the optic nerve cells literally just starve and die.
It's historically been a massive brick wall in medicine.
Because you can't just, send a microscopic repair crew into a cell to fix billions of tiny engines. Historically, diseases caused by broken mitochondria were considered basically untreatable.
Completely untreatable. I mean, for decades, if the primary energy source of a tissue, like an optic nerve or a beating heart, was compromised at that organelle level, doctors were basically helpless.
But a new paper suggests that maybe we don't have to fix them. Maybe we can just replace them entirely.
Yeah, it is a radical shift. It really is.
So welcome to today's deep dive. We are exploring a, honestly, completely groundbreaking 2026 paper published in Nature by Ayupov, Roska, and their colleagues.
It's a fascinating read.
The goal for our deep dive today is to unpack this revolutionary biological platform they've engineered. It's called MitoCatch.
Right, Mito.
And the premise is staggering. It's a highly specific delivery system that lets scientists physically transplant healthy, fully functioning mitochondria directly into specific sick cells.
And they aren't just doing this in isolated lab dishes either. They are moving toward complex living biology.
Which is wild. So if you're listening, we are going to break down exactly how they pull this off. We'll look at the viral inspiration behind it, the 3 different targeting strategies they use, how they prove the organelles actually survive, and finally, a real-world medical application rescuing those blind optic nerve cells.
Yeah, the sheer ambition is, well, it's what really stands out here.
Yeah.
Because, mitochondrial transplantation isn't like a brand new concept.
Right.
But the historical approach to it was deeply flawed.
Because before this, the old method was basically just taking a bunch of healthy mitochondria and dumping them onto sick cells.
Right.
Just hoping some of them would find their way inside.
Yeah, exactly.
It sounds like flying a cargo plane over a city that needs emergency supplies and just kicking crates out the back door without parachutes.
That is actually a highly accurate way to visualize it. It had incredibly low efficiency and absolutely zero targeting.
Just totally random.
Right. You were just flooding an area and relying on the cells to passively take them up. But the researchers behind MitoCatch, they realized that nature already solved this exact targeted delivery problem, billions of years ago.
Oh, viruses.
Exactly, viruses. They looked at viruses as the ultimate microscopic delivery vehicles.
Because viruses don't just bump into things randomly. They have keys.
Precisely. A virus uses viral coat proteins that bind to very specific cell surface receptors. It's a literal biological lock and key.
Like a zip code?
A highly targeted zip code. And the researchers hypothesized, well, what if we could give isolated, healthy mitochondria their own viral-style zip codes?
Which would solve the whole dropping crates without parachutes problem.
Exactly. It solves the targeting and the efficiency problem in one go.
Okay, so that brings us to their first major engineering feat, the strategy they call MitoCatch-C.
Right.
And if you're listening and trying to keep track, just remember the C here stands for catcher on the cell. So how exactly do you build a custom catcher's mitt on a living human cell?
Well, they started by genetically engineering the target cells. They used standard laboratory cells, HEK293-2T cells, and also induced human neurons, which they call IH neurons.
Okay.
And they modified these cells to display a highly specific binding mechanism on their outer surface. Basically an anti-GFP nanobody.
Wait, I want to pause there because nanobodies are fascinating. They're basically miniaturized versions of antibodies that we get from camelids, right? Like alpacas or llamas.
Yeah, exactly. Llamas and alpacas.
And because they're so tiny, they're super stable and easy to use as biological tools.
That's the exact reason bioengineers love them. So they took this alpaca-derived nanobody, which is designed to grab onto only one specific thing, green fluorescent protein, or GFP.
Okay, so it only hunts for green glowing stuff.
Right. And they fuse that nanobody to a transmembrane domain called PDGFR-beta.
So that PDGFR-beta is basically like the anchor that embeds the grappling hook firmly into the cell's outer wall.
Exactly. The cell is now sitting there, reaching out with a hook that will only catch something glowing green. That's the lock.
Got it. So what's the key?
For the key, they prepared the donor mitochondria. They took healthy mitochondria and fused that green fluorescent protein, the GFP, to an outer membrane protein called OMP25.
Okay, OMP25.
Yeah, OMP25 naturally sits on the outer surface of mitochondria anyway, so it acts as the perfect mounting bracket for the green bait.
Oh, that's so clean. So you have a perfectly matched system. The target cell is covered in anti-GFP locks, and the donor mitochondria are covered in GFP keys.
Exactly. And when they introduced these two engineered components to each other,
it must have been way better than the old cargo drop method.
the efficiency just skyrocketed. They recorded a 1,300% increase in fluorescence compared to control cells.
Wow. Wait, 1,300%.
Yeah. We are looking at a capture rate where 94.5% of the target cells successfully grab the mitochondria.
That is a stunning metric. But okay, I have to play devil's advocate for a second here.
Sure.
Because grabbing something on the surface is not the same as actually using it safely. I mean, our bodies have immune cells, right? Macrophages that constantly grab things just to pull them inside and destroy them in the lysosome, the cellular garbage disposal.
Right. Endocytosis, regular cells do it too.
Exactly. So if I'm looking at this critically, how do we know the cell isn't just treating these glowing green mitochondria as like a foreign invader, just wrapping them in a digestive bubble and dissolving them for spare parts?
That is honestly the pivotal hurdle of any intracellular delivery system.
Yeah.
To prove they weren't being digested, standard fluorescence wasn't enough. They had to use transmission electron microscopy.
To really zoom in.
Right, paired with a specialized dark stain called MiniSOG.
MiniSOG, that sounds like a tiny pair of wet shoes. What does that stand for?
It stands for Mini Single Oxygen Generator. It's a brilliant imaging tool. When you hit MiniSOG with specific wavelengths of light, it generates reactive oxygen species.
Okay.
And that polymerizes a chemical in the sample into this dark, highly electron-dense stain.
Oh, I see. So by attaching MiniSOG to the donor mitochondria, they could put the cells under an electron microscope and literally track these dark-stained packages to their final resting place inside the cell.
Exactly. And when they looked at those high-resolution images, the mitochondria were beautifully intact.
They weren't dissolved at all.
Not at all.
Yeah.
The images showed them sitting safely inside the neurons. And more importantly, they maintained their cristae.
The cristae, those are the highly folded inner membrane structures, right?
Exactly. Those folds are essential for energy production. If the mitochondria had been digested, those delicate folds would be the very first things to disintegrate.
Okay, let's take a quick conversational checkpoint for you listening, just to lock this in. So the main problem with older therapies was a total lack of a delivery address.
Right.
Ayupov and Roska solved this by borrowing from viruses. They engineered a custom grappling hook MitoCatch C, catcher on the cell, and armed the healthy mitochondria with the green bait. They proved it gets inside, and the electron microscope proved it stays physically intact.
Perfectly intact.
But I'm still stuck on one physical problem.
What's that?
Even if they are intact, if a cell pulls them inside via endocytosis, they are still physically trapped inside a tiny membrane bubble, an endosome.
Right, the entry vesicle.
Yeah, and to actually power the cell, they need to break out of that bubble and swim freely in the cellular fluid, the cytosol. Did they find a way to prove they actually escaped the bubble?
They did. And the mechanism they used is, I'd argue, the most elegant piece of molecular engineering in the entire paper.
Oh, really?
Yeah. They created a spatial detection system using a destabilized nanobody. It's called DGBP1.
Destabilized meaning what? It naturally falls apart if left alone.
Exactly. Under normal conditions in the cellular fluid, this specific protein is totally unstable. It quickly degrades and effectively vanishes.
Okay.
But the researchers fused this disappearing protein to a blue fluorescent protein called TAG-BFP.
Wait, I think I see where this is going. If you have a blue glowing protein attached to something that constantly degrades, the cell shouldn't glow blue at all. It's basically invisible ink.
That is the baseline state, yes, invisible ink. However, this destabilized protein has one very specific quirk. If it comes into direct physical contact with green fluorescent protein...
The bait on the mitochondria.
Exactly. If it touches the GFP, it instantly binds to it, and the moment it binds, it stabilizes. It stops degrading.
Oh, that is incredibly clever. Let me walk through the geometry of this for the listener.
Go for it.
So the donor mitochondria are covered in GFP. If the cell pulls those inside but traps them inside a little endosome bubble, the blue protein floating in the main cellular fluid can't touch them. The bubble is a physical barrier.
Right.
So the blue protein keeps degrading and the cell stays dark.
You have the logic perfectly. The only possible way for the blue protein to physically touch the green mitochondria is if the mitochondria successfully break out of that endosomal bubble and enter the open cytosol.
And if they escape, the blue protein binds to them, stabilizes, and boom, the area lights up blue.
It is a definitive binary test. If it glows blue, the package has been opened and the contents are free.
That is so cool. So what did the numbers look like for this test? Did they actually escape?
The results were definitive. In their target neurons, 70.3% of the cells lit up blue.
Wow. So a massive majority.
Yeah, and the overall fluorescence of that blue protein was 1,200% higher in the cells that captured the mitochondria compared to the controls. It was undeniable proof. They survived the journey, escaped the garbage chute, and were fully exposed to the cellular ecosystem.
Okay, so once they're swimming freely in that cellular ecosystem, do they just like float around aimlessly or do they actually get to work?
Oh, they actively integrate. They used live imaging techniques to literally watch these newly arrived organelles navigate the cell.
Like tracking them on a map.
Yeah, they traveled along the neurites, those long branch-like extensions of the neurons, moving at the exact same speeds and patterns as the cell's native mitochondria.
So they know exactly where they are.
Right. And perhaps most importantly, they observed them undergoing the natural cycles of fission and fusion, meaning they were actively splitting apart and merging together with the existing network.
Wait, so they don't just operate as like isolated rogue batteries. They physically blend their outer membranes and share their internal contents with the host's original power grid?
Yes. The researchers used a red dye, DS-Red2, to label the inner matrix of the native host mitochondria.
Okay.
By watching the green donor mitochondria merge with the red native ones, they confirmed the physical sharing of materials. The transplanted organelles were totally functionally assimilated.
Okay, let's do another quick recap. Memory checkpoint number 2.
Sounds good.
So they use this ingenious blue light trick, the invisible ink, to prove the mitochondria escape the cellular garbage chute. Then they watch them travel down neural highways and literally fuse and share parts with the host's original mitochondria. It's like dropping a new engine into a running car and it just immediately hooks up to the transmission, starts sharing oil with the old engine, and gets right to work.
That's a perfect analogy.
But okay, if I'm a doctor looking at this paper, I still see a massive clinical bottleneck.
The engineering step.
Right. MitoCatch C requires us to genetically engineer the target cell to grow that catcher's mitt. But if a patient comes into my clinic with a failing heart or degenerating optic nerves, I can't easily gene edit billions of their sick cells inside their living body to grow custom alpaca nanobodies, right?
No, you definitely can't.
So how do we take this out of the petri dish and put it into a real patient?
That's the exact limitation the researchers anticipated, which brings us to the next evolution of their platform, flipping the script.
Flipping the script.
If clinical application prevents us from modifying the patient's cells, the only solution is to engineer the therapy itself to do the hunting. They call this approach MitoCatch M.
Okay, M for putting the grappling hook directly on the mitochondria.
Correct. They return to that outer mitochondrial protein we talked about, OMP25. But this time, instead of attaching the green bait, they fuse the specific nanobodies directly to it.
So the donor mitochondria themselves are now fully armed with the targeting binders.
Exactly. To test this, they equipped the mitochondria with binders that specifically hunt for a natural protein called CD31.
Why CD31 specifically? Like, what kind of cell are they hunting?
CD31 is heavily expressed on the surface of endothelial cells, the cells that line the interior of our blood vessels.
Oh, I see.
By arming the mitochondria to look for CD31, they successfully directed them to enter human umbilical vein endothelial cells. They just leveraged the natural biological markers of the cell as the targeted zip code. No editing of the target cell required.
That immediately makes this so much more clinically viable. But reading through the paper, they actually took it one step further, right? With a third variant that feels like the ultimate, most adaptable version of this tech.
Yes, MitoCatch Bi.
Bi stands for bispecific.
Right, because rather than permanently engineering the cell or permanently modifying the donor mitochondria, they created a standalone, free-floating recombinant protein that acts as an intermediary. It has two distinct binding domains.
It's basically molecular double-sided tape.
I love that analogy. It's exactly that. One side of this protein tape is designed to latch on to a natural, completely unmodified protein found on all mitochondria,
Yeah.
like TOMM20.
Okay.
And the other side of the tape is designed to latch onto a specific natural receptor on the target cell, like CD71.
The elegance of that is just hard to overstate. I mean, you don't have to gene edit the donor and you don't have to gene edit the patient. You just mix healthy, off-the-shelf mitochondria in a solution with this double-sided tape. The tape sticks to the mitochondria, priming them to instantly stick to whatever natural cellular marker the other side is looking for.
Exactly. The adaptability of this bispecific approach is what allowed them to finally move from isolated cells in a dish into a living organism.
Because they tested this in vivo, right?
They did. They injected this MitoCatch bi mixture directly into the eyes of living mice.
Oh.
And guided by that double-sided molecular tape, the healthy mitochondria successfully navigated the complex biological environment inside the eye and integrated directly into the targeted retinal ganglion cells.
All right, time for recap number 3. Just think about the progression here.
It's fast.
Yeah. We moved from engineering a specific target in a lab dish to putting the grappling hook on the mitochondria, that's MitoCatch, and then finally creating a modular double-sided tape MitoCatch bi that actually works inside the eye of a living mammal.
Right.
But there's one final frontier we really need to cover, because proving the delivery works is one thing. Proving it actually rescues a dying human cell from a genetic disease is another entirely.
Absolutely. And to cross that final threshold, the researchers apply their system to a severe human pathology.
Yeah.
Liver hereditary optic neuropathy, or LHON.
LHON. This brings us right back to the disease we mentioned at the very beginning of the deep dive, the painless, rapid loss of central vision in young adults.
Yes. So LHON is driven by a very specific mutation known as MT11178A.
Okay.
This mutation occurs in the ND4 gene, which resides entirely within the mitochondrial DNA.
So because the mutation is isolated to the mitochondria, the cell's main nucleus is completely healthy.
Precisely. The core blueprints of the cell are fine. But the power plants are producing toxic reactive oxygen species, and they're failing to generate enough ATP, the energy currency of the cell.
So the optic nerve cells are structurally fine. They're starving to death.
Exactly. So the team took induced human neurons, those IH neurons, derived directly from a patient suffering from LHON.
So these are real sick human cells carrying that exact MT1111078A mutation.
Right. And they applied the MitoCatch system to these dying cells.
So what did the rescue look like at a genetic level? I mean, did it actually work?
They flooded the patient-derived neurons with healthy wild-type mitochondria. To verify the transfer, they used single-cell RNA sequencing.
To look at the transcripts inside.
Exactly. When they analyzed the interior of the treated sick cells, they found them repopulated with healthy, wild-type ND4 genetic transcripts.
So the new organelles were actively running their uncorrupted genetic code inside the sick cell.
Yeah.
But okay, having the healthy transcripts is basically just having the right blueprints. It doesn't necessarily prove the factory is actually producing power, right? I imagine they had to put these cells under some kind of extreme stress to prove the new mitochondria were actually keeping them alive.
They did, and they utilized a brilliant biological stress test for this. It's called a galactose survival assay.
Galactose, like the sugar.
Right. To understand why this test is so definitive, you have to look at how cells normally consume sugar. Human cells strongly prefer glucose.
Sure.
When bathed in glucose, a cell can actually cheat its energy requirements through a process called glycolysis.
And glycolysis happens in the regular cellular fluid, right? It bypasses the mitochondria entirely.
Exactly. It's a backup system.
Ah, I see. So if the sick cells are sitting in a petri dish full of glucose, they might survive even with broken mitochondria because they're just running on the backup generator.
You nailed it. So the researchers completely removed the glucose from the environment and replaced it with galactose.
And what does that do?
When a cell tries to process galactose, it cannot use glycolysis to achieve a net gain of energy.
Oh. So you are systematically shutting down their backup generators? You're forcing the cell into a state where it must rely 100% on oxidative phosphorylation, the specific energy process inside the mitochondria.
It becomes a pure binary outcome of survival or death. If your mitochondria are broken, you die.
Oh.
And when the untreated LHON patient cells were placed in the galactose medium, they died. Their mutated mitochondria simply could not handle the energetic load required to process the galactose.
And the cells treated with the MitoCatch system.
The treated cells not only survived the galactose stress test, but they demonstrated massive, statistically significant increases in both basal respiration and ATP-linked respiration.
That's incredible.
The transplanted healthy mitochondria had completely restored the sick cell's ability to breathe, generate power, and survive under lethal metabolic stress.
Okay, final recap here. LHON causes blindness via broken mitochondrial DNA. The MitoCatch system delivered healthy mitochondria into real patient neurons. And the galactose assay proved the cells regained their ability to actually breathe and survive.
Exactly.
I mean, the philosophical shift here is just incredible to think about. When we talk about genetic diseases today, the conversation is like almost entirely dominated by CRISPR and gene editing. Right?
Oh, absolutely.
We talk about going in with molecular scissors and snipping out the bad code, but this is completely different. We aren't doing a software patch on the broken DNA. We're doing a massive hardware swap.
Right.
We're overwriting the faulty code by just flooding the environment with billions of healthy organelles that carry their own uncorrupted blueprints. It's wild.
It really expands the therapeutic horizon in a profound way. Because if the organelle itself is fundamentally broken, the most efficient solution might not be to painstakingly repair it, but to systematically replace it. And, the researchers emphasize that MitoCatch is not just an eye treatment.
It's a platform technology.
Yeah.
Simply, it's a platform.
Because if you just change the zip code on that double-sided tape, you could theoretically target any tissue in the body.
That's the ultimate promise here. By swapping the specific nanobody or binding domain, you could direct healthy mitochondria to ischemic tissue fighting for survival after a stroke.
Wow.
You could target failing cardiac cells in a patient with severe heart disease, or degenerating neurons in Parkinson's or Alzheimer's disease. The delivery mechanism remains exactly the same. You just change the address on the envelope.
We started this deep dive talking about a city with a failing power grid. What Ayupov and Roska have engineered isn't just a replacement turbine. They've built like a fleet of autonomous, highly targeted delivery vehicles that know exactly which city needs help and how to seamlessly plug straight into the failing grid.
It's revolutionary.
But as we wrap up, I think this technology forces us to ask a really, really strange question about human biology.
It does. It brings up a fascinating dilemma regarding genetic identity.
Well, mitochondria are entirely unique among our cellular components because they maintain their own distinct circular DNA, right? It's completely separate from the 46 chromosomes in your main nucleus.
Oh, right. And mitochondrial DNA is passed down exclusively from the maternal line.
Exactly. It traces an unbroken line back through your mother, her mother, and so on. So imagine a near-future scenario where a patient with severe heart failure is treated with a systemic injection of MitoCatch.
Okay.
We transplant billions of healthy mitochondria from a generic donor into their cardiac tissue. Those heart cells will now be entirely powered by and running on someone else's maternal genetic code.
That's wow.
From a purely biological standpoint, that patient becomes a cellular chimera.
It really challenges our definition of self at the microscopic level. I mean, if the very engines keeping your cells alive, dictating their energy, their stress responses, and their survival, if they belong to a stranger, how does that change the genetic definition of you?
It's a massive question, and it's one that the medical ethics and philosophy fields will really have to grapple with as organelle therapy moves closer to human trials. We are learning how to mix and match the fundamental building blocks of life in ways that were previously just unimaginable.
That is a phenomenal thought experiment to leave you with today. If you take anything away from this deep dive, let it be the sheer ingenuity of borrowing viral mechanics to perform cellular hardware swaps. Next time you take a deep breath or, feel a burst of energy, give a little bit of credit to the billions of ancient, semi-independent power plants keeping your metropolis running. Thank you for listening, and keep questioning the incredible biology happening inside you right now.